All right. Can you guys hear me? Welcome back. Happy Wednesday. Let's start by doing a quick review. I'm going to go ahead and present to you guys. You don't have to write any of this down. We're just going to talk through a couple of review slides, and then we're going to jump into making a protein today based on what we learned yesterday. So right now it's starting to present my screen. You guys see DNA versus RNA? Thumbs? Good. Thank you. So we understand the structure of DNA. We know that there is a language difference, right? We're using thymine and DNA. We're using uracil and RNA. The tricky part comes along when we're recognizing which type of RNA we're using. Because as you guys have found out, there are more than just one type of RNA. There's messenger, which is known as the codon. There's transfer RNA, which is known as the anticodon. And then there's the ribosome itself. Okay? And so, again, when we're going from codon to anticodon, we're speaking the same language. However, when we're actually reading DNA during transcription, we're going from thymine language to uracil language. So our triplets are all going to have thymines, but instead of a thymine for our codons, we're going to be using uracil. Okay? So please don't forget that. Now, the two charts that you can use to figure out what amino acid is being added to your protein is here in the presentation, but I also uploaded it for you on the Google Classroom. So please open up one of them right now. You can open up the sundial one if you like, or this one. So yesterday we used the sundial. Today I'll be using this chart. You have to be comfortable using either or. And so if you open up this one and put it off to the side in one of your tabs, you can click back and forth to see how I'm reading it. So yesterday we looked at this DNA segment. Each one of these three base segments is called a triplet. This triplet then gets transcribed into a codon. A codon is a three base segment of messenger RNA. This whole strand is messenger RNA, but each of these three bases is its own codon. So we have DNA triplet 1, 2, 3, triplet number 4. This would be codon 1, codon 2, codon 3, codon 4. Does that make sense to everybody? Okay. For every codon, there is an anticodon. So we've got codon 1, anticodon 1, codon 2, anticodon 2, and so forth. Remember, we're just trying to get our letters straight first. Once we get all of our letters straight, the language is correct, then we have to keep in mind that we're not going to be able to build a protein unless we find our start codon. And I had you guys write down the start codon letter yesterday, but I'm going to type it in again today. You can't start making a protein until you see the start codon AUG. So be careful as I'm giving you guys your DNA segments and you're using the transcribing process to put it into RNA language. You're also kind of in the back of your mind looking for AUG. Ah, here's AUG. Start codon. Beautiful. All right. So keep that in mind. This right here is not going to be anything, right? Because it's not a start codon. So if that's not a start codon, then there's not going to be an anticodon. But once we find AUG, then we can start building our protein, right? And then at the end, when you get to a stop codon, which is UGA process of making a protein. And then whatever you had built will end up being released, packaged up by your Golgi apparatus, and then released either out of the cell or used in the cell. Okay? Does anyone have any quick questions before we start our first problem? Anyone need clarification on anything? Can go switch cameras. You're on my presentation. I'm going to go ahead and go back to the chart. So what you guys should be seeing right now is the chart on one screen and my overhead on the other. Are we good? Thumbs? Okay. See, so this is the part that I want you to put in your notes. Now, you don't have to draw this DNA strand at the top. What I'm showing you here is that we're starting inside the nucleus, right? This is the nucleus, and there's DNA inside the nucleus. We are going to use DNA polymerase. Excuse me, not DNA polymerase. I misspoke. RNA polymerase to start building our messenger RNA strand from reading this little segment that we're going to open up. So let's see, 1, 2, 3, 4, 5, 6, 7. So let's get this RNA, and this is what we're looking at after we open it up. So copy down these triplets, 1, 2, 3, 4, 5, 6, 7, into your notes right now. And again, if you need to pin it to make it bigger on your screen, go ahead and do that. Easier to read. So if we're going to make a protein now, what's our first step of protein synthesis? What's step 1? What's it called? Somebody yell it out.
Transcription.
Yes, transcription. Transcription is step 1. So we're going to take and start making messenger RNA, and this is called, each one of these little 3 base things is a codon. So what's our first codon for ATA? We're going from DNA language to RNA language. I'll do the first one. So the first codon is going to be UAU. Do you understand why it's UAU? Can't use thymine, remember? Based on that, what's our next codon? So the codon for the triplet GCC is going to be CGG. Rose petal, what's the codon for TAC?
AUG.
Yep. Very good. Roy, what's the codon for AAA?
UUU.
UUU. Michael K., what is the codon for GTC?
CAT.
CAG, right? So GTC would be CAG. Make sense, everybody? Kylie, what is the codon for AAT?
Is it UUG?
For AAT with UUA, right? Does that make sense, everybody? Francisco, bring us home, man. What is the codon for ATT?
UAA.
UAA. So this whole thing right here is messenger RNA. Each one of these is a codon. Give me a thumb if that makes sense to you. All right. So that was step 1, transcription. What's step 2?
Translation.
Translation. So this is actually transfer RNA anticodons. So for every one codon, we said there was going to be a complementary anticodon. But guess what? They speak the same language, so we don't have to worry about T's, right? We're just going to be using U's here on out. Now, the idea behind this anticodon is the anticodon is responsible for bringing an amino acid. And so when we talk about amino acids, they're going to be pulled by the transfer RNA anticodon. Think of the anticodon as being like the delivery boy, right? Where it's going to bring, this is Amazon. It's going to drop the amino acid package off at your doorstep, depending on what codon you paid for. So let's look at and figure out what our anticodons are. So this is our codon. What's the anticodon for UAU? AUA, correct? Please write that down. Andrew, what's the anticodon for CGG?
GCC.
Johnny, what's the anticodon for AUG?
UAC.
UAC. Reese, what's the anticodon for UUU?
AAA.
You got it. Nathan, how about the anticodon for CAG? Nathan, you there, man? Nate Dogg, you got me? All right, I know he's there. I know he's moving. He's alive. He's waving. That's a good sign. All right. So, Nate, what do we have here? CAG is the codon. What's the anticodon for this guy? G. U. C. Anna, how about the anticodon for UUA?
AAU.
Very good. And then bring us home, dear Kaylee. UAA has the anticodon.
AUU.
Beautiful. Now, remember, I said each one of these guys should be bringing an amino acid like an Amazon delivery truck. And so now we have to figure out exactly what amino acid we have to be bringing picture. Are we amino acid is appropriate? The blue, the red, or the purple? Looks like somebody said red. That is correct. The red codons are used to read the chart that we have here. So the first codon is UAU. U-A-U. The answer for UAU is tyrosine. But that's not a start codon. And because that's not a start codon, we cross it out because we can't start making a protein yet. Right? We have to wait till we find AUG to start making a protein. The next codon reads CGG. CGG, that's arginine. That's not a start codon. So we get to cancel that guy out. The next codon in sequence is AUG, AUG, which is methionine. And that is our start codon. So the amino acid methionine is always going to be your first amino acid in your protein. So we're starting to make a protein now. Little check mark there. The next codon in sequence is UUU. Andrew, what's the amino acid that codes for UUU?
Is it cysteine?
Nope. U,
Oh.
the third letter.
What is that? Phenylalanine? Phenylalanine.
Yeah, phenylalanine. That's a tough one. Phenylalanine. Say that to yourself at home, phenylalanine. And then guess what? You get to bond those together. We're building a protein. We're making a long chain. So now we're going to put a little bond here, attaching methionine to phenylalanine. Making a protein, gang. CAG. CAG. Where's Marta? Marta, are you there? Hello, dear. CAG, kiddo. What's the amino acid for CAG?
Glutamine?
Yes, very good. Glutamine. Everyone say glutamine to yourself at home. GLUT I'll put, and then we join it to the first two. So now we have 3 amino acids in a row. We got a nice chain going to make our protein. What's the next one? Let me see. Victor Garcia. UUA.
UUA is leucine.
UUA is leucine. This guy's a pro. Leucine. Go ahead and attach that. So now we have 4 amino acids. Boom, 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 boom in a chain. We got a protein going. UAA is the last anticodon. Aya, UAA, kid, what does that look like on our chart? UAA. It's a stop codon. So this guy right here is actually a termination factor. Right? So we don't add anything here. This doesn't really bring in amino acid. It's just telling it to stop making a protein. So what we did here, we just made one protein. We attached our amino acids together. We have a nice long chain here of protein. And this third step, what's this third step called? Step 1 is transcription. Step 2 is translation. And as we added amino acids to make a long chain, this is called what?
Elongation.
Yep. Yeah, Nathan, I couldn't hear you. I don't know, maybe other people could, but I could not hear you. Did anybody else hear him? No? Okay. if you guys, like if I call on you and your mic is not working, you can always just type in your answer too. I mean, I would prefer that you speak, obviously, if your mic works, but I get it. I knew you were there, Nathan. So man, now you've cranked out a protein. Now you've performed protein synthesis. This is a big deal, gang. Who has questions? Anybody need clarification?
I have a
What, bro?
So I got a little behind, but where did the methionine come from? Because was I supposed to use the chart? Because that's not on the chart. So how
Yes.
do you know that was meth?
I know that that was methionine
Okay.
because in order to read and know which are amino acids, we have to use the codons to do that.
Mhm.
And AUG code name, why we were talking about earlier, in my other classes, somebody tried to use the DNA code up here to read the chart, and they didn't get anywhere really fast because there's no T's, right? The charts don't have any T's. It's all U's and A's and G's and C's.
Yeah.
So don't use the DNA, use the codon. Some people try to use anticodons, and that'll screw you up too, because those anticodons are not on the chart. Only the codons are on the chart. So you're going to always start with AUG. That's our start codon. That's methionine. UUU is phenylalanine. CAG was glutamine. UUA was leucine. And then this was our termination factor. This was our stop codon. So that just tells us, hey, we're done. Stop.
Okay, I used the wrong chart. I got it. Thank you.
What were you looking at?
I was looking at the circular one.
That chart is the same thing, kid.
But it's,
Wow.
well then how am I getting the wrong? Why am I getting the wrong thing still?
So here, let me present. So take a look at the sundial. Here's the sundial. AUG was methionine. AUG is methionine.
Okay, that's
Does that make sense?
Yeah.
And then UUU, here's this first one. UUU is phenylalanine. And then CAG is the third. CAG is glutamine. And then UUA is leucine. And then UAA is stop. Does that help? So whether you use this one or you use this one, they should give you the same answer. Thumbs from everybody? Okay. Now, what did I say about those charts? You have to know how to read both of them, right? Because you're not going to know which one I'm going to put on there, Rose Petal. If I put the one that you're a little, you're not really good at, I don't want you to do poorly because you don't know how to use that one. But I think you know how to do it now. So you should be good. Questions? All right, gang. Homework is posted. It's another simulation on what we just did right now. Tomorrow we'll get more practice in, talk a little bit deeper on what this stuff is all about. And then you should expect a quiz on Friday. And then the test will probably be Tuesday, I think. At least that's what I'm hoping for. So go over your notes, people. Go over your notes. Who knows the DNA song? Anna, stop it. You better know it, people. Roy, did you commit it to memory? A little bit. I'm telling you right now, Colton, you're going to get throat chopped if you don't know how to play that song. You got like 12 guitars in the background. You probably have a keyboard over there. So, Smeda? No? DNA song? No? Son of a gun. All right, gang. You got a lot of work to do. Get in your notes. Make sure you do the homework by midnight, and I'll see you guys tomorrow. Have a great day.